Okay, so hi guys. So for this topic, we will be discussing the basic practices in the different sections of the clinical laboratory. So we will be discussing first the section of clinical chemistry, and then the clinical microbiology and the clinical microscopy. Okay, so that is for the first part. Okay, for clinical chemistry. Okay, so this section or yes is also known as the clinical biochemistry, clinical pathology, medical biochemistry, or pure blood chemistry. But actually, guys, this is the most commonly used term, that is clinical chemistry. So this CC, okay, so or CC, the area of pathology that is generally concerned with the quantitative analysis of substances found in the blood or serum. Okay, why is it termed blood or serum? Okay, so in this part, mostly in clinical chemistry, the commonly used specimen that is the serum. Okay, so serum, that's the most commonly used sample in clinical chemistry, but plasma may also be used depending on the test that is utilized. But in our country, mostly of the machines are quality controlled using the serum as the specimen. Okay, so we also receive whole blood as a sample in clinical chemistry, particularly for HbA1c test. Okay, so most of the time you will receive serum unless you have a test request that is HbA1c. So the HbA1c test, it actually measures the sugar component of that is present in hemoglobin. So it is a long-term measure of the sugar content. Okay, so for diabetics, it measures almost 2 months of the sugar, okay, in the blood. So HbA1c, why does it use whole blood? Because the hemoglobin A1c that is found in the red blood cells. And therefore, you cannot use serum because the analyte that you are looking for is not present in the serum. Rather, that is present in the red blood cell. Okay? So, your sample for HbA1c that is whole blood, that is particularly placed in a tube, in a purple tube with the anticoagulant of EDTA, okay, or the EDTA. So, not only the serum or the whole blood, okay, but samples also such as urine, spinal fluid, pleural fluid, they are often analyzed, but these are very rare, in rare occasions. Just like for urine, that is in creatinine clearance, okay, so in in my experience, I haven't process the spinal fluid and pleural fluid in the clinical chemistry sections yet. Okay, so that's it. So when we say analyte, this is the substance for which analysis are performed. Okay, so when we're referring to the analyte, we are referring to the substance that is being tested. Okay, so tests are often performed in groups of individual tests. They are called panels. So the panels provide a broad database demonstrating multi-system involvement. So tests are performed with any kind of body fluid, but mostly on serum or plasma. Okay, but mostly in clinical laboratory, we use the serum. So we have panels. So that is groups of individual tests. So large laboratories accept samples for up to 700 different kind of tests. Okay, even the largest of laboratories rarely do all these tests themselves, and some need to be referred to other laboratories. These large areas of tests can be further subcategorized into specialties of general or routine chemistry. Okay, so even for tertiary laboratory, they do not offer Okay, they do not offer all of the analytes. So, which means that they do not test all of the analytes. Okay, so for example, in clinical chemistry section, they do not test all of the analytes under clinical chemistry. Okay, so in these cases, a laboratory can send a sample to a different laboratory which offers the test. Okay, so for example, a clinical laboratory, which is a tertiary, does not have a test for hemoglobin. Yes, for what do you call this one? Let's say total iron binding capacity. Okay, because these are rarely tested. Okay, so they may refer it to other laboratories which offer the test. Okay, so pwede yon. Like for example, in uh, what do you call this one? Some tertiary laboratories do not have tests for a certain analyte in their lab, and they refer that to CBMC. Okay, something like that. So we have here endocrinology. So in clinical chemistry, guys, it has so many branches. So it has many topics. Okay, even in the subject of clinical chemistry 1, clinical chemistry 2. Okay, so these are some of the topics that will be discussed. Endocrinology, so that is the study of hormones. Okay, immunology, the study of immune system and antibodies. But actually, guys, most of this, the endocrine and immunology analytes, so these are mostly tested in immunology and serology section. So pharmacology or toxicology, that's the study of drugs. Okay, so we differentiate serum. Okay, so serum, that is a yellow watery part of the blood that is left after the blood has been allowed to clot and all blood cells have been removed. This is most easily done by centrifugation. Okay, so you remove the fluid portion of the blood. Okay, you separate it from the cellular elements, which packs the dense blood cells and platelets to the bottom of the centrifuge tube, leaving the liquid serum fraction resting above the packed red cells. For example, here. Okay, so this is a plasma. Okay, so let's discuss first plasma. Okay, plasma, essentially the same as serum, but is obtained by centrifuging the blood without clotting. Plasma contains all the clotting factors, including fibrinogen. So actually, the main difference between a serum and plasma is that serum has no fibrinogen because the fibrinogen has been used in the clotting process. Serum doesn't have an anticoagulant, okay? So there is no chemical that prevents the coagulation process when you utilize a serum. That's why it doesn't have a fibrinogen, okay? So it does not also contain any anticoagulant. In plasma, okay, so actually, mostly they are the same in the sense that plasma and serum are taken from a whole blood. Okay? The main differences between these two are, of course, the presence of fibrinogen in plasma. And, of course, the plasma did not undergo the clotting process and it contains an anticoagulant. Unlike for serum, okay, you place this in a tube with no anticoagulant so that the clotting process will be done. Okay? So essentially the same as serum but is obtained by centrifuging the blood without clotting. Plasma contains all the clotting factors including fibrinogen. And this fibrinogen is absent in the serum. Okay, so we have here the composition of blood. So when you draw blood, so actually this one, guys, if this is a tourniquet, so it is misplaced because that must be 3 to 4 inches away from your puncture site. Okay, so we have here veins. So if you draw blood, okay, you will have a plasma or serum depending on the tube where you will place the whole blood. If you put the whole blood into a tube that has no anticoagulant, definitely you will have a serum after the centrifugation process. But if you put a whole blood in a tube with an anticoagulant, you will obtain plasma after the process of centrifugation. Okay? So, kapag may anticoagulant, you will have plasma. Pag walang anticoagulant yung ginamit na tube, you will have serum. Okay? So, we have here, so since this is termed as plasma, most likely this is, it was placed in a tube with an anticoagulant. So, plasma, that is the uppermost part, okay, which makes up the 55% of the whole blood. And then that is followed by the buffalo coat, which contains the platelets and the WBC. And then what do you call this one? What you found at the bottom of a centrifuge blood with an anticoagulant is the erythrocyte or the red blood cell. Okay? Okay, another difference between plasma and serum is that, okay, in serum, guys, the formed elements will be mixed together. Okay, the platelets, the WBC, okay, and the red blood cells will be formed together. Okay, so that is for the part wherein serum separates from the formed elements. In the Buffy coat, okay, so, sorry, in plasma, okay, so again, plasma is the fluid portion of the whole blood, okay, which contains the fibrinogen because of the presence of an anticoagulant. Here kasi in this whole blood which contains the plasma, once you mix this one, okay, they can be mixed together, okay? They can be mixed together, okay? And aside from that, the Buffy coat or the platelet and WBC will be separated from the erythrocyte. Okay, so that is for the plasma, okay, or the whole blood which contains the plasma. On the other hand, for serum, okay, once the formed elements are clotted, hindi mo na siya pwedeng ma-mix. Okay? Kasi nag-form na together yung platelets, WBC, and the red blood cell. And aside from that, there is no clear separation of Buffy coat and the red blood cell. Okay? So, in a sample with serum, okay? Blood collection. So, the basic rule when drawing blood is to collect enough to run a test 3 times. Okay, while ensuring that you do not take so much blood that you compromise the health of the patient. So, which means that in drawing blood or getting a sample from the patient, you get enough sample, okay? So that if you need further tests on that particular sample, you won't need to draw blood again. Kaya nga sabi dito, enough to run a test 3 times, okay? It doesn't mean, guys, that for example, the test is glucose, okay? It doesn't mean that you will run the test 3 times, but it means that you must have an excess of the sample so that in cases wherein additional test must be done, you already have enough sample without getting blood again from the patient. Okay, in blood collection, you must avoid hemolysis. When you say hemolysis, that is the destruction of red blood cells resulting in the release of hemoglobin and cellular contents into the plasma or serum. Or when you say hemolysis, that is as simple as the lysis of the red blood cell. Okay, so we have here, so this is the biconcave dish shape of a red blood cell. So this is a red blood cell, and this is a hemolyzed red blood cell, wherein the hemoglobin are released into the fluid portion of the whole blood. Okay, so like for example, this one, the number 1, that is a normal. Okay, so there is no sign of hemolysis. Okay, so this is the, what do you call this one? This is the formed elements. So for number 2, so we can see here slight hemolysis. Okay, so we have here different degrees of hemolysis, and number 4, that is heavy hemolysis. Okay, so that is reflected by the reddish color of the serum. Okay, so that is reflected by the reddish color of the serum. That is hemolysis. Okay, so there are different causes of hemolysis or lysis of the red blood cell. Okay, so if you use too small needle with respect to vein size, that may lead to hemolysis. Okay, so when you use gauges, when you say kasi gauge of the needle, it refers to the size of the hole of the needle. Okay, so if you use a small needle to draw blood, Okay, so that may lead to hemolysis. Okay, so using a needle smaller than gauge 23, okay, or 23 gauge. So this one again, it refers to the diameter of the hole of the needle, okay, in a syringe or in evacuated tube system. So over and under filling of tubes. So this refers to tubes with an anticoagulant. So when you say overfilled, okay, wait, I will just copy an image so that it's not hard for you to imagine what do you mean by over and underfilling of tubes. Okay, so overfilling, guys, for tubes with anticoagulant, there is a level wherein you must only fill the tube in that certain level. So, for example, this is a 3 ml at the tube, so you must only, okay, fill the tube up to this level. Okay, if you put more blood, so that is overfilling of the tubes. So, pag sumobra dito sa line na to, okay, mas madami, that is overfilling. Pero kapag mas maliit naman, hindi umabot yung dugo. For example, you're only able to get a sample up to this area. Okay, up to this level, hindi mo to naabot. That is the underfilling of tubes. So, overfilling and underfilling may also lead to hemolysis. Okay, so vigorous mixing or shaking of tubes. So guys, in different tubes, we have a recommended number of immersion. Okay, so hindi pwedeng i-shake mo siya ng maraming beses. Okay, so hindi mo siya pwedeng ma-overmix or ma-undermix. If undermixing of tubes with anticoagulant may lead to clotting. Kasi you were not able to mix that properly. But over mixing or vigorous mixing, that may lead to hemolysis. Like for example, in EDTA, so the recommended inversion for EDTA tube, that must only be 8 inversions. Okay, so if vigorous mixing or over mixing of tubes, that may lead to hemolysis or lysis of the red blood cells. Okay, another using a small needle with a large vacuum tube. So I'll just get another image for this one. Okay, so this one, so this is the Vacutainer tube system or the evacuated tube system, which medtech use also for extracting blood. So this one, if you use a very small needle, okay, with a large vacuum tube. So for example, you use a very small high gauge of needle. The relationship kasi between a gauge of a needle and to the size of the diameter is inversely proportional. Kapag mas mataas yung number ng gauge, okay, again, the gauge refers to the diameter of the hole of the needle. So, pag mas mataas yung gauge niya, ibig sabihin, mas maliit yung hole. Pero kapag mas mababa yung gauge niya, mas malaki yung hole. Okay, inverse relationship of the diameter of the hole, yes, and the number of the gauge. So, if you use a very small, yes, a very small needle, so it refers to the hole of the needle. Malit yung butas ng needle, and malaki yung vacuum tube na ginamit mo. Like, for example, this one, or 5 mm of tube, okay? And use a very small hole of a needle that may lead also to hemolysis. Okay, kasi ang laki ng pressure. Okay, mataas yung pressure para humugot ng blood and yet maliit yung hole ng needle mo. So that may lead to lysis of red blood cell. Okay, using a small needle to transfer blood from syringe to tube. Okay, so this is especially common. This type of hemolysis is common if you use a needle to extract blood. Syringe. If you use a syringe to extract blood, okay, and then you transfer that into an evacuated tube by just puncturing the. I know it's really hard for you to imagine this yet. Okay, but I hope you can still get it. Like for example, okay, in the collection process, you use a syringe. Okay, you use a syringe, and then you put the sample into a evacuated tube. Okay, So guys, instead to avoid hemolysis, you must remove the cup, okay, and transfer what is present in the syringe to the tube, okay. Tatanggalin mo yung cup and then ilalagay mo dun yung sample. However, there are cases wherein you will just puncture the tube, okay, to transfer your sample from the syringe to the tube. So, in those cases, that may also lead to hemolysis. And again, aggressive mixing or vigorous mixing. Parang naulit lang. Okay, hemolysis can interfere with laboratory results due to several processes. Okay, so if the analytes are normally present in greater amounts inside the red cells than in plasma, the leakage of analytes from the red cells may cause false increase in the amount of analytes measured. So this may occur when measuring the levels of potassium, creatine kinase, and alanine aminotransferase. But these are just some, okay? Konti lang to relative to the analytes that are increased when there is hemolysis. Okay, so let's try to explain what this means. Okay, so guys, if hemolysis happens, take note, the analytes that some of the analytes that are present in the serum, their concentration is higher inside the red blood cell. So if the red blood cell are lysed, okay, nasira yung red blood cell, anong mangyayari sa mga analytes na nasa loob ng red blood cell? Lalabas sila. Saan sila pupunta? Pupunta sila sa serum or sa plasma. In that case, what will happen? That will lead to increased levels of those analytes. Tataas yung level nila. Is that normal? Of course not. Okay? Kasi that will lead to false increased levels of analytes. Okay? So, like potassium, creatine kinase, and alanine aminotransferase. So, if there is hemolysis, especially if that is heavy hemolysis, these analytes, but not only these analytes are increased, these are just examples. Okay? So, these analytes, they increase in levels falsely. Mali yung pagtaas ng level nila dun sa serum or plasma. Kasi, meron lang hemolysis. Kapag walang hemolysis, iba yung madedetermine na value compared kapag may hemolysis. So, anong ginagawa ng hemolysis? It increases the levels of the analytes. Okay? So, kagaya ni potassium, tataas. Okay? So, if the analytes are normally present in greater amounts in plasma than in red cells, the analyte will be diluted by the lysing of the red cells, causing a false decrease. Sodium and chloride can both be decreased. Okay, so anong ibig sabihin nito? Guys, not all analytes are increased once there is hemolysis. There are also analytes which decreases in value because of hemolysis due to dilution. Okay, take note, inside the red blood cell, meron ding fluid yan. Okay? Meron din siyang fluid. So, kapag nag-hemolysis, anong nangyayari sa plasma? Nagkakaroon din ng dilution. Okay? Kapag yung analytes, mas marami, normally, kapag walang hemolysis, pag mas marami yung analytes sa plasma or serum kapag walang hemolysis, at kapag nagkaroon naman ng hemolysis, anong mangyayari doon? There will be dilution. And in that case, what will happen to the level? Magde-decrease siya. Okay? Like si chloride and si sodium. Okay? Hemolysis may lead to false decrease in value of sodium and chloride. But that is inverse in case of potassium. Kasi mas maraming potassium ang nasa loob ng red blood cell. Mas konti yung potassium na nasa plasma or serum. So if there is hemolysis, the level of potassium is increased. But if there is hemolysis for sodium and chloride, their levels may be decreased. Okay? Color interference, tinting of the serum, pink or red, can cause false increase when using a spectrophotometer. Okay? So if you use the method of spectrophotometer to measure the analyte, okay, that may also lead to false increase in value. So hemoglobin, just like hemoglobin, bilirubin, protein, so are a few of the analytes affected by hemolysis if you measure their values using a spectrophotometer. So guys, in laboratory, okay, or in extracting blood from the patient, you must avoid hemolysis because that may lead to false increase or false decrease of the value of the analyte. Pwedeng tumaas or pwedeng bumaba. So, false increase or false decrease in values may lead also to misdiagnosis of the physician. So, kapag pinases mo yung heavy hemolyzed sample, for example, okay, so pinases mo yung heavy hemolyzed, and then, yung level ng potassium niya is sobrang taas from the normal range. So, that may lead to misdiagnosis of the physician and administering unnecessary medicine to the patient just because you gave a false result. Okay, so be careful with that. Sometimes, red cell constituent can react with analytes, causing a false decrease. So, this may occur when testing for carbon dioxide, thyroxine, and insulin. So, there are constituents in the red blood cell that may react with carbon dioxide, thyroxine, and insulin, leading to a false decrease. Okay, kagaya ng sodium and chloride. So, kapag nagkakaroon ng hemolysis, bumababa sila. So, pwede din daw bumaba ang carbon dioxide, thyroxine, and insulin. Hemolysis can cause increased turbidity when using the refractometer to determine blood protein levels. Okay, so if you use refractometer, okay, in measurement, that leads to increased turbidity naman. Okay, another, avoid hemolysis and avoid lipemia. So, lipemia, okay? So, another interference that can adversely affect test results is the lipemia. It can also enhance hemolysis by increasing red cell fragility, okay? So, lipids present in a specimen scatter light and can cause either an increase or decrease in values depending upon the analytes being evaluated. So, just like for hemolysis, lipemia can also lead to a false increase or false decrease in values of the analytes that are being measured. Okay, so how does it differ from hemolysis in terms of physical appearance? So, in hemolysis, guys, what you see is plasma or serum that has a reddish color, okay, or red pinch in color. May pagka-reddish siya or pinkish. That is for hemolysis. Okay, so in lipemia naman, it looks like whitish or milky in color. So these samples, these are lipemic, okay? So the serum, they are lipemic. So this is a normal serum, okay? Ma'am, how can you classify if that is serum or plasma depending on the tube or the color of the tube? So this one, this is red. So this is serum. So this is serum. But for this one, since this is EDTA, so it has an anticoagulant, therefore this is plasma. Okay? So lipemia, so it can be cloudy or whitish in color, unlike for hemolysis, which is pinkish or reddish in color. Okay, so this is a normal color of the serum. Okay, straw-colored fluid, or yes, straw-colored or light yellow. Okay, so that is for the normal serum. So because red cells are in aqueous phase of the blood, lipids may dilute their concentration. Okay, so it can be minimized, however, by fasting patients prior to blood collection, by ultracentrifuging the sample, or by using polymers to precipitate the lipids out of the sample. So in clinical chemistry, guys, to address this one issue of a sample that is being lipemic, so we require our patients to fast depending on the test that is being done. So for example, glucose. So we require them to fast for 8 to 10 hours. Okay, so but it depends on the laboratory you are working with. So some laboratories require 6 to 8 hours of fasting. Other requires 8 to 10 hours of fasting for glucose alone. But if there is another test, that is glucose with lipid profile, okay, so some um, uh, uh, requires patients to fast for uh, 10 to 12 hours, okay? So, while some, they allow 10 to 14 hours. So, it depends on the laboratory. Okay? So, fasting, what do you mean by fasting, guys? So, you require your patient not to eat, okay, anything or drink or minimize their drinking of water. Okay, so no coffee, no sweets, no teas, no food, okay? So, that is fasting. Okay, so fasting for depending again on the test, but if that is glucose, pwedeng 8 to 10 hours. So the timing that is started from the last meal. Okay, so kung kumain yung patient at 9 p.m., so 10 p.m. yung first hour, okay, na nag-fast siya. So that is for fasting. Okay, so electrolytes. So these are the analytes that are commonly tested in clinical chemistry section. So we have electrolytes, so these are enzymes. Okay, so as you can see guys, in naming, most of the enzymes, they end with the ASE. Okay, later ASE phosphatase, kinase, dehydrogenase, terminase, amylase. Okay, so those are enzymes. So protein, so we have here total protein. So albumin, that is the protein which has a the highest concentration of protein found in the serum. Siya yung, kumbaga, pinakamalaking proportion ng protein na nakikita sa ating dugo. So that is albumin. So organic compounds, glucose, urea, uric acid, creatinine, acetone, cholesterol. Okay, so guys, in lipid profile pala, so we have there 5 tests under lipid profile. So, ano yung mga kasama sa lipid profile? We have the cholesterol, okay, or total cholesterol. We have the VLDL, very low density lipoprotein, VLDL, low density lipoprotein, HDL, high density lipoprotein, and the triglyceride. Okay, so those are the 5 analytes that are measured in lipid profile. So, 5 yun. A more useful classification of analytes organizes analytes by organ or diagnostic profile. So, we have, okay, so we have this one. So, profile type, renal. So, this, guys, so this analytes, okay, may reflect the kidney function. Kaya nga, renal or kidney function test. Okay, ito minsan tinatawag na kidney function test. Liver function test, ito. Sa liver function, these analytes, they reflect the activity of the liver. So, these analytes, they reflect the activity of the kidney. So, you may measure these types of analytes or these kinds of analytes when you want to assess how the kidney is functioning. Okay, same with the others. Sa heart naman, so we have there CPK, LDH, troponin T, CPK-MB, myoglobin. But the most commons are the troponin. Okay, the troponin test, which is commonly requested in cases of myocardial infarction or heart attack. Itong troponin I and troponin T, these are stat requests. When you say stat request, this test request must be tested immediately. Agad-agad na dapat niya-test. So, upon receipt of the sample for troponin test, dapat yun na yung priority mong i-test kahit meron ka ng ibang mga sample na na-receive. Okay, and thyroid test. So, dyan yung T4, thyroxine, free thyroxine, thyroid uptake, triiodothyronine or the T3, and the thyroid stimulating hormone. And actually, guys, this is a thyroid test. Okay, so these are commonly tested in immunology and serology section. Okay, but maybe it depends on the classification of the, or it depends on the machine that is used by a laboratory. Okay? So, hypertension. So, the this one, the lipid profile, cholesterol, HDL, LDL, VLDL, triglyceride, they are requested to assess the hypertension or high blood pressure. Okay, that is commonly known as the high blood pressure, hypertension, as well as for renin. Okay, so chemistry system. So these methods that can be used to determine the chemical composition of a certain fluid. So, kumbaga, these are the methods to test for glucose, HDL, okay, albumin. So, we have there the dry reagent strip. The dry reagent strip requires the composition of a color change on the reaction strip with the color chart. So, this is most commonly used as a quick screening test, and its accuracy is only low to moderate. Okay, some reagent strips include machines that read the color change, and these are more accurate than the color change measured by looking at the color. So we have there the dry reagent strip. Another is wet and dry chemistry. It utilizes spectrophotometer to mechanically measure color change and are much more accurate than the dry reagent strip. So the reagent is the difference between the two systems. Most chemistry systems today use dry reagents which are mixed with a liquid specimen. So spectrophotometer, it uses the Beer-Lambert's law. So Beer-Lambert's law, the Beer's law to measure concentration. So this law states that the amount of light absorbed by a solution varies with the concentration of the colored solute. Light is shown through a specimen of cuvette, and the amount of light transmitted is recorded by a photocell. This is then converted into the amount of substance in the sample. So spectrophotometer, that will be further discussed in your clinical chemistry 1 when you reach second year, second sem. Okay, so commonly submitted specimen for clinical chemistry, mostly serum. Pwede rin naman plasma. But serum is more commonly tested or more commonly submitted than plasma, and whole blood. Okay, whole blood, that is for what test again? HbA1c. Okay, hemoglobin A1c. So that is for the clinical chemistry section. So we proceed with another section, that is clinical microbiology. Okay, guys, so let's proceed to another section of the clinical laboratory, that is microbiology section. But for some hospitals or some laboratories, they refer this as the bacteriology section because they only process specimens or tests that are related to bacteriology. So, yung tinetest na lang nila ay mga bacteria, okay? So, there are 4 subsections: bacteriology, mycobacteriology, mycology, and virology. Okay, so depending on the microorganism that is intended to be tested. Okay, so guys, though parasitology is under microbiology, the tests that are commonly requested to assess parasitism are under clinical microscopy. Okay? So, yun yung nangyayari sa ating clinical lab. Kahit ang parasite ay under ng microbiology, in the actual clinical laboratory testing, under sila dun sa clinical microscopy section. Okay? So, microbiology, it involves the identification of bacteria and fungi on specimens that are received. Okay? So, common samples that are submitted are blood, body fluids, stool, tissues, and swabs. So this, guys, this is a biosafety cabinet. So we process samples inside the biosafety cabinet so that the samples are not contaminated by the environment, and so as to provide protection to the medical laboratory technologies or those who will process the sample. Okay? Gumagamit tayo ng biosafety cabinet para protektahan yung ating mga sample against contamination and to protect also the worker from acquiring the organisms that are present in the sample. Okay? So, in microbiology, this is the microscopic visualization of microorganism after staining, isolation and identification of bacteria, either that is aerobes or anaerobes, and fungi using varied culture media and biochemical tests. So, we have two types of bacteria depending. Actually, not only two types, there are others, okay? So, but here, we classified bacteria based on their growth requirement in oxygen, okay? So, kapag aerobes, these are bacteria that survives or lives in the presence of oxygen. Pag anaerobes naman, they survive or these bacteria survive in the absence of oxygen. So guys, if you remember from our previous discussion, we mentioned the commonly used materials or equipment and machines. Okay, so we mentioned the use of culture media, and again, this culture media, they provide the nutrient growth for the bacteria to grow. Bakit nga ba natin kailangan ang culture media in the bacteriology section? That is to shorten the time for the bacteria to multiply. Okay? Para isang araw pa lang, kasi diba normally, the bacteria that are present in the sample, we don't see them. Okay? Because of their very small amount, and of course, their very small size. But when you use culture media kasi, sobrang napapabilis yung pagdami ng bacteria because you are giving them the nutrients that they need to multiply. So we have here an example of culture media. So when you place a specimen that has a bacteria, okay, so after 24 hours, they multiply. Okay, so in a very fast manner, wherein you can see colonies already. Itong mga to, colonies yung mga yan, and those colonies contains millions of bacteria. Okay? So, through culture media, pwede mo nang makita yung isang colony ng bacteria. Millions of bacteria within just 24 hours. So, without using culture media, matatagalan bago dumami ang bacteria mo. Okay? So, at times, antigen typing and antibacterial susceptibility testing is also performed in the microbiology section. So, the antigen typing, so this is commonly done in advanced microbiological laboratories. Okay, so this antimicrobial susceptibility testing, this is commonly done, okay, even in ordinary or in a microbiology section. Okay, so preparation of culture media. So these are routine activities as well as we do not prepare stains, instead we just purchase them. Okay, so culture media can also be purchased, but of course, if you want to lessen the expenses in your laboratory, you may prepare them yourself. Okay? Okay, so we proceed to the test commonly done in, oh my gosh, in micro biology section. So we have culture test. Actually, this one, what is commonly ordered is the culture and sensitivity testing. Okay, so dalawa yun, culture and sensitivity testing. However, for the purpose of discussion, let's discuss them separately for you to understand it better. Okay, so for the culture test, a bacteria culture test can help find harmful bacteria in your body. During a bacteria culture test, a sample will be taken from your blood or other parts of your body and will be inoculated in culture media to determine whether microorganisms have invaded the patient. The type of sample depends on the location of the suspected infection. Actually, in the culture test, it is as simple as getting a sample from the suspected site of infection and then putting that in a culture media. So if there are growth in the culture media or if there is a growth in the culture media and that is what do you call this one? The growth is enough to be considered as positive for infection, they will proceed to susceptibility testing. Okay? Guys, it doesn't mean na kapag may nag-grow sa culture media, meron ka ng infection. There are cases wherein merong contamination na nangyayari, or there are cases wherein may growth siya pero hindi sufficient yung growth para sabihin merong infection. Okay? So this one, that is, you either submission of sample and then inoculating the sample into a culture media. So actually, guys, this culture media, this is referred as the sheep's blood agar. Bakit siya sheep's blood agar? Because the culture media contains a sheep blood, okay? Literal na merong dugo ng sheep sa agar na to. Okay? So, this one, it has growth, it is positive for growth of bacteria, and actually, that is already a heavy growth. Okay? Heavy growth, marami. Meron kasi tayong light growth, medium, and heavy growth of bacteria. Okay? So, this is a culture media with a heavy growth. Okay, guys, in sensitivity testing, this is also called sensitivity analysis. Okay, so it helps your doctor find the most effective antibiotic to kill an infecting microorganism. Okay, so actually, this one, guys, this is sensitivity testing or susceptibility testing. Okay, so if the sample is positive for bacterial growth, okay, merong growth na nakita after the culture test, they will proceed to susceptibility testing to be able to determine the types of antibiotics that are still working against the bacteria. Okay? Kung ano yung mga antibiotic na pwede pang gamitin sa bacteria. So, in the process, you will get a small sample of the bacteria. Okay? So, you mix that with saline solution. Then, that will be inoculated in Moller-Hinton agar or Moller-Hinton cake. So, the disc will be placed. So, what are these discs? Antibiotic discs. So, these antibiotic discs are placed on the agar plate and then that will be incubated again. When you say incubated, ilalagay mo siya sa incubator for 18 to 24 hours. And then, after that time, you will get this one and check kung meron bang susceptible na antibiotic and you will indicate those in the result. So, yung mga yan, yung mga clearing na nangyari dyan, those are zone of inhibition. Okay, yan yung tinatawag na zone of inhibition. Okay, so yan yung i-measure and then yan yung i-report. Yan yung basis kung pwede pa bang gamitin yung antibiotic dun sa bacteria na na-isolate. Okay? So that is for sensitivity or susceptibility testing. So we have here different samples. As you can see, guys, okay, so the different antibiotic reacts differently. Okay? Yung iba, maluluwang yung zone of inhibition. Yung clearing na nangyayari. Okay? So, let's just use the other image for reference since that is clear naman. So, for this one, okay, so the antibiotics, since these are different antibiotics, the bacteria react differently with the different types of antibiotic. There are antibiotics which produces larger zone of inhibition. So, yan yung nangyayari na clearing. And the larger the zone of inhibition, it means that it is more, the bacteria is more susceptible. Mas namamatay yung bacteria kapag yun yung ginamit mong antibiotic compared sa mga antibiotic na konti lang yung zone of inhibition. Okay? So, another commonly requested test that is the Gram stain. So, it is a test that checks for bacteria at the site of suspected infection or in certain body fluids such as blood or urine. there are two main categories of bacterial infection based on gram stain. Okay, we have the gram positive and the gram negative. So, how do you classify if a bacteria is gram positive or gram negative? It actually depends on the color after the gram staining. Kapag purple siya or blue, but mostly that is purple, that is gram positive. Kapag gram negative naman, red or pink yung color ng bacteria. So, in gram staining, it utilizes four different reagents. Okay? So, meron kang primary stain, that is the crystal violet. Okay? So, that is the crystal violet. And the mordant, that is the iodine. Another is alcohol, which is used for decolorization. And the counter stain, which is the safranin. So, bias, remember the acronym bias, V. So, that is crystal violet. So, yung V doon yung V, yung bias. Yung V sa bias. That is crystal violet, followed by iodine, and then alcohol, and then safranin. So, the use of those reagents that is crucial. So, these reagents are used to determine the gram classification. Okay, kung gram positive siya or gram negative. And this is the order, chronological order of how you utilize the reagent. Mauuna yung crystal violet, the primary stain, the iodine, which is the mordant, the alcohol, which is the decolorizer, and the safranin, which is the counter stain. Okay? So, bakit purple, violet, or blue yung ating gram-positive bacteria? Okay? So, most likely, it's not blue. It's actually, dapat ang mas lumalabas na color doon is purple or violet. Okay? So, the gram-positive bacteria retains the color of the primary stain, which is the crystal violet. Kaya siya nagiging violet or purple at the end of the staining process. Kapag gram-negative bacteria naman, ang nire-retain niya na color is that of the safranin. Kaya nagiging red or pink yung bacteria na gram-negative. Okay? So, alalahanin yung may yun. So, another test, that is the acid-fast bacilli test. These are usually ordered for people with symptoms of active tuberculosis. The test look for the presence of AFB bacteria in the sputum. So, the positive color reaction in the AFB stain, that is the presence of bacilli which has a red or which has a red color or pink in color. So, these are AFB. Okay? So, this is indicative of Mycobacterium tuberculosis infection. Kasi what causes TB is the Mycobacterium tuberculosis. Okay? So, that is for the AFB test. Another test, that is the KOH test for Candida albicans, which is also known as the potassium hydroxide. Okay, the potassium hydroxide, that is the reagent that is used for the KOH test. Okay, so that is a quick, inexpensive fungal test to differentiate dermatophytes and Candida albicans. Symptoms from other skin disorders like psoriasis and eczema. Okay, so that is for the potassium hydroxide. So actually KOH, that is the what do you call this one? Symbol, okay? Chemical symbol for potassium hydroxide. Okay, so we have morphologies of bacteria. We have here bacilli. Kapag bacilli, okay, elongated. Okay, pahaba. Pag cocobacilli, so we have here, that is in between cocci and bacilli. And we have the cocci kapag spherical siya or bilog-bilog. Okay, so pag bilog-bilog, that is cocci. Okay, so quality assurance and quality control is done also in microbiology, infection control. So this must be practiced. Biosafety must also be observed, and proper waste disposal so as not to contaminate the environment and so as not to inflict harm to those who are working outside the laboratory. Okay? So commonly submitted specimen, urine, blood, stools, sputum, and other samples. Okay? So that is for the microbiology section. So we proceed with the clinical microscopy section. Okay. So we proceed with clinical microscopy section. There are two major areas in clinical microscopy, that is the routine and the special exam. Okay? Routine urinalysis and special exam of urine and other body fluids. Okay, so what are tested or yes, tested in routine urinalysis, not also in routine urinalysis but also in special tests. That is the gross appearance, microscopic appearance, and the biochemistry. Okay, yung gross or physical appearance na nakikita mo sa sample, microscopic kapag tinignan mo siya sa microscope, and biochemistry kapag meron ka ng mga test na ginawa. Okay? So in routine urinalysis, that is, a urinalysis is a simple test that looks at a small sample of your urine. It can help find problems that need treatment, including infection like urinary tract infection and kidney problems. Sa special examination naman, that is, analysis of urine and other body fluids. May mga other body fluids, cerebrospinal fluid, pericardial fluid, pleural fluid, peritoneal fluid, synovial fluid, semen. Okay, so those are other, but there are many other specimens. So tests done on other body fluids like cell count in synovial fluid and body fluid, sperm count, and others, these are classified under special examination. Pero kapag routine urinalysis, urine yung i-analyze mo. Okay? So, another, stool or routine fecalysis. So, this stool or routine fecalysis, these are commonly done to assess for the presence of parasites. Okay? So, here, the patient must pass 3 stool samples collected at 2-day intervals. Pero guys, pwede lang din naman na isang sample lang. Depende kasi yan sa test request ng doctor. So, this increases the chances of detection of hard-to-find parasites like Charcholambia, hookworm, Ascaris lumbricoides, okay? So, this one, the stool or routine fecalysis, that is intended to see if a patient has parasitic infection. Another test, that is the fecal occult blood test or the FOBT, that is a lab test used to check stool samples for hidden or occult blood. Occult kasi, that means hidden, okay? So, occult blood in the stool may indicate colon cancer or polyps in the colon or rectum. Though not all cancers or polyps bleed. So, here kasi in fecal occult blood test, it will test for the presence of blood. Okay? So, hindi lang yung blood na nakikita by the naked eye, but as well as the blood that has already been destroyed, okay, due to oxidation. Okay? So, assessment for the presence of colon cancer or polyps. So, that is for the fecal occult blood or testing for the presence of hidden blood in the stool. So, samples commonly submitted, that is urine for routine urinalysis, stool for the routine fecalysis, and other samples. So, that is for the clinical microscopy section. So, this ends the part 1 of our discussion for the different sections in the clinical laboratory. So, meron pa pong part 2. So, thank you for listening. Bye!